Hello dear students, in this lecture we will discuss about the your syllabus with respect to experiment to prove whether DNA has a genetic material. So in this regard we will discuss about the experiment of, we can say, or the name of an experiment is nothing but the Griffith experiment. Here, before going to DNA, already we have discussed about the structure and molecular properties of DNA. And now we are moving towards the different characteristics and different processes which are carried out in every cells with respect to either this prokaryotic cell or in case of eukaryotic cell. But scientists of that time, they didn't know whether the genetic information is transferring from one generation to next generation is through either it is DNA, either it is RNA or polysaccharide or protein or other sort. So that information which is transferring from one generation to next generation, by which route they didn't know. So that to prove that information is moving or the genetic material is stored or genetic information is stored on DNA, that experiment or that evidence was given by the Griffith, and that experiment, to carry out that experiment, he used two different types of strains of bacteria, that is R type of strains and ES type of strains. So let's discuss whatever experiment he has carried out and what are his findings. So the early work of British bacteriologist Frederick Griffith in 1928 on the transfer of virulence in the pathogen, that is Streptococcus pneumoniae, gave first evidence of DNA as a genetic material. While working on the Streptococcus pneumoniae, he given his evidence of his experiment that virulence factor, it is nothing but the ability of that particular organism to cause a disease, or you can say pathogenicity, is transferring from one organism, either it is from R to S or S to R, through some sort of protein, RNA or DNA. To carry out that experiment, he set up an experiment, and for that he selected two strains of bacteria, that is Streptococcus pneumoniae. For that he selected two strains of Streptococcus pneumoniae for his experiment, and that experiment is nothing but the Griffith experiment, we can say. R type of strain, or we can say it is nothing but the rough type of strain, we can say, when grow on a petri plate, it produces or it shows a rough type of colony appearance, or hence we have he noted that strain as a R type of cell, as it produces rough type of colony appearance, and the bacterium is non-virulent in nature and non-capsulated, and that particular bacteria is non-virulent and he could not produce a capsule around his cell. So properties of R type of cell, or we can identify that particular R type of cell by means of it shows a rough type of colony appearance on the media when we can grow on that particular bacteria on media. It is non-virulent type of bacteria and non-capsulated or non-capsule form. Another type of bacteria which he have taken, it is yes type of strain. When he grown that particular bacteria on a petri plate, it produces smooth type of colony or smooth appearance on the media plate, and that bacteria is virulent in nature and it is forming a capsule. That means two types of different strains, whether they are belong to the same genus, R type of strains and S type of strain. One is virulent, another is non-virulent. One is capsule former, another is non-capsule former, and one shows rough type of colony and one shows smooth type of colony. So after that he carried out the experiment. So he carried out his experiment on a mice, or you can say it is in laboratory mice, you can say all type of experiment are carried out on that mice. So firstly he taken a yes type of cells, that is smooth type of cells from the virulent bacteria which is virulent in nature, and injected the yes type of cell into a healthy mice, and he found that mice becomes ill and dead. And after the death of that organism, he isolated that particular yes type of organism from the dead mice. That means that cell or that organism is virulent in nature, and when injected into healthy mice, it produces particular disease, and that mice become ill and dead, and yes type of one is outside. Later on, he injected the R type of cells, that is rough type of colony, we can say, or rough type of bacteria into a healthy mice, and after that he found that the mice didn't show any side effect into his, and the mice become or remain healthy, or no symptoms develop with respect to that yes type of cells. After that, he third step, or he carried out the third type of experiment. It kills cells of yes type of cells. That means he killed the yes type of cells by heat, and he injected that mice into a healthy mice, and he found that mouse remained as it is. No infection was observed on that mice. Later on, he used all type of cells plus yes type of heat killed cells. So what happened? He used another fourth type of experiment, all type of cells and heat kill yes type of cells, and injected that mice, and he found that mice dead, as that result was observed in type of first type of experiment, and yes type of cells are recovered from the body of the mice. Here in fourth step, he injected the heat kill cells of yes type of cells, as he has injected into third step, and he found that mice remain as it is. But when he injected heat kill yes type of cells and R type of cells in combination into mice, he found that mice become ill or dead, and he recovered the yes type of cell from that dead mice. That means, as he injected the heat kill cells of yes type, but he recovered live yes type of cell after the death of that mice. So from that experiment, he concluded that Griffith concluded that the R type bacteria have taken up something from the heat kill yes type of cells, and he called that component or that substance which R type of cells taken from the yes type of cells as a transforming principles, which allow them to transfer into smooth coated bacteria and become virulent. So after his experiment, and he concluded, or he draw his conclusion that something is transferred or something is taken up by the R type of cells which are non-virulent in nature from the heat kill yes type of cells. Something is transferred from the virulent factor to non-virulent bacteria. That's why that non-virulent bacteria becomes virulent, and he caused a disease to particular mice, and the death of that particular mice is happen, and that substance which is transferring from yes type to R type, he called that as a transforming principle. He didn't know whether it is transferred, whether it is RNA or DNA from the organism, but he called that as a transforming principle. But not sure what being transferred from yes type of cells to R type of cells. So it was the first evidence given by Griffith about something is transferring from one organism to another, and that R type of cells become virulent. But he not sure about whether it is RNA, whether it is DNA, or whether it is protein or polysaccharide which is transferred from yes type of cells to the R type of cell. So he called that as a transforming principle. It is all about the experiment of Griffith with respect to evidence of DNA as a genetic material. Next part we will discuss in next video.